The goal of this video lecture is to provide a brief overview of the biological or medical approach to conceptualizing psychopathology, including this approach's definition of a mental illness, some of the key concepts that are important for understanding this approach, and the interventions that are emphasized by this approach. From a biological or a medical model perspective, mental illnesses are viewed as being comparable to physical illnesses. So what this means is that emotional disorders, or psychopathology, are conceptualized as disease states that have an underlying biological cause and define symptoms. So this assumes that there's an underlying deficit or dysfunction in the underlying physiological processes that lead to normal behavior, and that this deficit or dysfunction is responsible for the symptoms that we observe that characterize mental illnesses. Many of the key concepts for understanding the biological approach are related to understanding the physiological processes that might be related to the development of psychopathology. One important area of research from a biological or a medical perspective is understanding potential genetic contributions to psychopathology. So an emphasis in studying the relationship between genes and mental illness and psychopathology, there are a number of questions that researchers ask. So one is whether we can identify particular genes that are associated with increased or decreased risk for mental illnesses. But in many cases, when we're talking about genetic contributions, we're not talking about single genes. Instead, mental illnesses tend to be what's called polygenic, which means that multiple genes and interactions among multiple genes are related to the development of psychopathology. It's also important to consider that there's not necessarily a one-to-one relationship between the genes a person has and the symptoms that they experience or whether or not they have a mental illness. So there can be a difference between a person's genotype, so the genes that they inherit, the chromosomes that they have, and their phenotype, or the way that those genes are expressed. An important concept for understanding this is the concept of a gene by environment interaction. So a gene by environment interaction is a case where the expression of a gene, so the phenotype that is expressed, depends upon the environment that an individual is exposed to. So in this case, we're talking about genetic contributions to psychopathology or genetic contributions to symptoms, but that relationship isn't necessarily straightforward. The relationship between the genes a person has and the symptoms that they experience is going to depend on the environment that they are exposed to. So this is an example of a gene by environment interaction for a specific gene that we're going to talk about in more detail when we discuss depressive disorders. This is called the serotonin transporter gene. There are two different versions of this gene. There's a short version and a long version. And what we know is that having a short allele for this gene is a risk factor for depression, but it turns out that it's only really a risk factor for depression in the context of stressful life events. So as you can see from this graph, individuals who report having no stressful life events tend to report the same level of depressive symptoms regardless of which combination of alleles of the serotonin transporter gene they have. In contrast, as individuals experience more and more stressful life events, individuals who have one or two short alleles of the serotonin transporter gene report higher and higher levels of symptoms of depression compared to individuals who have two long alleles of the serotonin transporter gene. So we can think of the short allele as a genetic risk factor for depression, but without stressful life events, someone with the short allele is not at any greater risk for depression than someone who does not have a short allele. So in this case, the phenotypic expression of depressive symptoms depends upon the environment that someone with a particular genotype is exposed to. Another important mechanism to consider from a biological perspective is potential differences in brain structure. So here we are considering whether differences in the structure of the brain, which might include damage or degeneration to parts of the brain, could potentially be related to the development of symptoms of psychopathology. So this is an example from a study looking at individuals with psychotic disorders, specifically schizophrenia, that we'll take a look at in more detail when we talk about psychotic disorders. On the left, we have a healthy control patient with no mental illness. And on the right-hand side, you can see a structural view of the brain of an individual with a psychotic disorder. And you can see significant degeneration of the brain, and you can also see enlarged ventricular volume, which is also indicative of decreased brain matter. So here is a case where we can identify some potential differences in the structure of the brain. We might also be interested in talking about differences in brain functioning, as we might observe using our dynamic brain imaging techniques. So in this case, we might not be seeing structural differences in the brain, but we might be interested in understanding underlying patterns of brain activity that could be potentially related to symptoms of psychopathology. For example, are there specific regions of the brain that are more active in individuals experiencing specific symptoms, or that are potentially less active in ways that might help explain why that individual is experiencing those specific symptoms? We might also be interested in neurotransmitter functioning. So remember from introductory psychology that neurotransmitters are the chemical messengers of the brain. This is how neurons communicate between one another. So we can have a range of problems with neurotransmitter functioning that could be related to problems with behavior and experience. These might include an excess of neurotransmitters, so having too much of a neurotransmitter. We might also have deficits of neurotransmitters. And there are a range of ways in which these problems can occur. They can occur at the point of a deficit in production of a neurotransmitter, so if not enough of a neurotransmitter is being created. When we think about the synapse, we can have problems with the release of neurotransmitters into the synapse where not enough is being released. We can have problems with the reuptake of neurotransmitters. So remember that many neurotransmitters are sucked back up by the presynaptic neuron so that that neurotransmitter can be reused later to continue to communicate with other neurons. So you can have abnormalities in neurotransmitter reuptake. Many neurotransmitters are broken down in the synapse by various enzymes, so abnormalities in the breakdown of those neurotransmitters can result in excess levels of neurotransmitters or deficits in levels of neurotransmitters if you have enzyme activity that is too high. And you can also have problems on the end of the receiving neuron where you can have problems with the receptors and the binding of neurotransmitters. So there are a really wide range of potential dysfunctions in neurotransmitter functioning that can potentially be related to the development of symptoms of psychopathology. So from a biological or a medical perspective, our interventions are designed to target these underlying physiological deficits or dysfunctions. Many of our medical interventions are psychopharmacological interventions. These are medications that are designed to address the underlying pathophysiology. So these might include drugs like neurotransmitter agonists that mimic the effects of a specific neurotransmitter. They might include neurotransmitter antagonists, so drugs that block the effects of a specific neurotransmitter. They could also be drugs that work on the enzymes that are related to the degradation of neurotransmitters in the synapse. So for example, they might promote more efficient breakdown of neurotransmitters in the synapse to lead to decreased levels of a neurotransmitter in the synapse. Or they might inhibit the breakdown of neurotransmitters in order to result in higher levels of neurotransmitters available in the synapse. We can also use various technologies and interventions that are designed to directly alter brain activity or to stimulate particular regions of the brain. So one way that we can stimulate brain activity is through what's called electroconvulsive therapy, or ECT. In ECT, we directly apply electric current to the brain, typically through the temples, in order to alter brain functioning. So electroconvulsive therapy has been used as a treatment for treatment-resistant depression, so individuals whose depressive symptoms have not responded well to other kinds of interventions, and is also being explored as an effective treatment for a range of other disorders. So in this case, using electric current to directly stimulate the brain has been shown to address symptoms of various disorders. We can also apply electric current more specifically to particular regions of the brain. So one way that this is done is through a technique called deep brain stimulation. In deep brain stimulation, an electrode is implanted directly in the brain in a specific region of the brain, and that specific region is going to vary depending on the disorder that is being targeted and the patient's specific symptoms. and that implant is used to directly apply electric current over time on a regular basis to address an individual's symptoms. We also have some non-invasive ways of activating the brain and altering brain stimulation in ways that appear to be effective for reducing symptoms. So one relatively recently developed technology is called repetitive transcranial magnetic stimulation. This is abbreviated RTMS. And RTMS is a technology that uses magnets, so similar to the way that an MRI uses magnets to form an image of the brain. In RTMS, we use magnets to non-invasively alter the pattern of brain activation in a way that appears very promising for addressing symptoms, particularly of depression. So all of these different approaches to brain stimulation address symptoms of psychopathology by directly altering brain functioning. And finally, another medical intervention is that we might directly alter brain structure. So if abnormalities in brain structure are related to symptoms, psychosurgery and directly changing the structure of the brain to address these problems is one potential way in which an individual's symptoms could potentially be addressed.